cannot see the recording.
What? You cannot record. Do you want me to record? Oh,
No,
you do.
no.
You are.
No,
Okay.
I already started, I think.
Yeah, it is started. I can see it.
No.
Good.
Yes. Shall we start?
Yes, please.
In this lecture, I will talk about imaging multiple colors, either with fixed or adjustable filters. So the aim of this lecture is to give you an overview how imaging multiple fluorophores in a sample is achieved by using fixed or adjustable filters. And probably pretty much you are already familiar with this image. Here we have our objective, we have our sample, so it's we depict an inverted microscope, and under we have the objective, we have a cube which contains an excitation filter, the dichroic mirror, and the emission filter. Both excitation and emission filters have a fixed width. We have a light source with multiple wavelengths, and we have one sensor. So, of course, now the emission light coming from the light source will pass the excitation filter, the dichroic mirror, and will reach the sample. And the emission light coming from our sample will be reflected towards the sensor, passing an emission filter. And as I said, everything is fixed. It has a fixed width, which means that if we have several fluorophores, we will have to image them one at a time, sequentially. And each time we want to image a fluorophore, we will have to change this the cube, which is in the filter turret, for each of the fluorophores we want to image, which means that this will take some time. And we have, of course, a limited number of combination of cubes that we can have in the filter turret, and of course also limited number of combination we can use. So it's quite, although we have in this case I depict the camera, it will take time. In the most modern systems, the excitation and the emission filters are moved out. They are not anymore in a cube. They are in so-called excitation and emission wheels, and that increase the speed of imaging, because it's much the vertical excitation or emission filters are easier to they move faster, and of course you also get more freedom to combine the excitation and the emission filters, but nevertheless, they have a fixed width. Can we image simultaneously in one sensor? Previously, we sequentially one fluorophore at the same time, one fluorophore at one time. But yes, we can also image simultaneously if we have one sensor. And here I depict one way it can be done. Then you still have only one sensor, a camera. So the emission light that will come through, comes from the sample, will reach a set of different dichroic mirrors and fluorescent and emission filters and mirrors that will send the light. So these dichroic mirrors will send the light to different filters, after which the emission light that passes through the filter will be displayed equivalent on the camera chip in different quadrants. After that, imaging processing will take place. Then we will overlap all the channels. It's of course faster because now we simultaneously the signal is displayed and is on the camera. So we don't need to wait for any filter cube or vertical filters to move. But it needs a very, very accurate calibration of the system all the time, so we can overlap the images. So how can we image if we have several sensors? And in this case, either several cameras or several detectors. So there are several strategies the microscope manufacturers came up with. Either still, we can also use fixed width filters, even if we have several cameras and detectors, or we can use variable dichroic mirrors, or a combination of variable dichroic mirrors and variable emission filters, diffraction gratings or grids, or prisms. I will just take you through them, no technical details, just to have an overview how it works. So how can we image when we have several cameras? Well, obviously, as the name says, you can have several cameras, and the key here is to have so-called beam splitters. So you have the emission light coming, and you will have several beam splitters that will send the emission light. Everything comes at the same time from all your fluorophores to different split to different secondary beam splitters, which in turn again will send the light to via filters to the different cameras. The blue light will reach one camera, green, and so on and so forth. So it's simultaneous and is fast, but you need to buy to have several cameras. It's not a cheap setup. Or this can be also applied if, for example, that we are imaging with a spinning disk, and in this picture, I depicted how it works in the Opera Phoenix microscope or system, and basically here the excitation light will be projected on to opposite sides of the spinning disk or the spinning disk, and after which the light will be guided to your sample. In the same way, the emission light coming from the sample the emission light will be projected to the two different quadrants on the spinning disk or the micro lenses, and with the help of different beam splitter and dichroic mirror will be sent to each of the corresponding cameras. And it's again a very fast way to image, but of course you need several cameras. And of course the images are later combined in the software. So how about how it works if we have fixed width emission filters and several detectors, like for example the single point confocus have several detectors. And here I show an example how it works in the Nikon Nikon A1, the older version of the Nikon microscopes. So we have our lasers, we excite our sample, the laser will pass through the first dichroic mirror or the primary dichroic mirror and will reach the sample. Now the emission light coming from the sample will have to be directed to the detectors. Here we have 4 detectors, blue, green, for each of the collecting light from different blue fluorophores, green, red, and far red fluorophores. And first it will pass through the pinhole. And now we have another set of dichroic mirrors, so-called secondary dichroic mirror, which will again guide the line, guide the line towards each detector. via so-called emission filters. But these filters are also have a fixed width, and then will reach the detector. So, for example, the blue line, the blue light will reach the first secondary dichroic mirror here, and everything which is above 495 wavelength will go further. Everything will be reflected towards this detector via fixed width filter. So as you can see, the principle of working with fixed width filters and dichroic mirror are the same for wide field, multi-point confocal light sheet, and single point confocal system. The advantage in the single point confocal that if you don't have a bleed-through, you can actually image everything simultaneously. If you have a bleed-through, you will have to go sequential, or you can combine the different fluorophores. But we are still limited by the width of the emission filters. So how about if we are using variable dichroic mirrors? So can we do that? So different other manufacturers of microscope, they come up with other solution how we can image multiple fluorophores by using variable dichroic mirrors, and if we have several detectors. So here we have the emission light coming from the sample, which will pass the pinhole, and then we have so-called variable secondary beam splitter, size calls it this way, which will then either reflect the light via mirror to a first detector, and the rest of the light will pass and reach the second detector. And then we can move this variable secondary beam splitter, and then we do it again. And this is how, for example, it works in the LSM 700. Just a picture of how it looks inside, where we can see if we imagine the light comes, the emission light comes from the sample. We have the pinhole, and then we have the variable dichroic mirror, which will send the longer wavelength to one of the detectors. Here we have two detectors, one here, and the rest, the shorter, will pass through and will reach the second detector. In the LSM 800, we have two so-called variable secondary beam splitter, and we have three detectors if we consider the Airiscan detector too. But the principle how it works is basically the same. We adjust the light that will reach either each of the detectors by moving and using these very special dichroic mirrors. So the advantage of using the variable secondary beam splitter or dichroic mirrors is a very efficient separation of the emission light. All the portion of the emission spectrum is utilized, and we can achieve very fast simultaneous acquisition. We can also combine variable dichroic mirrors with so-called variable emission filters. And this is how it works, for example, in the Nikon IX, the newer Nikon version of the IX Nikon microscopes. We have here a combination of long-pass dichroic mirrors, short-pass dichroic mirrors. We have also 4 different detectors. And basically, what we do, we guide the line with the help of these dichroic mirrors. Some of them for the emission light coming from the sample will, depending on the wavelength, the longer wavelength will pass. We have a long pass dichroic mirror. The shorter via a filter, fixed filter, will reach the first detector. Everything else that passed will again encounter these variable dichroic mirrors. We can be moved with a few nanometers in the one direction or the other. And again, the light will be either sent to the second detector via linear variable filters, which can be tuned to a few nanometers, basically, and so on. And the light will further be will continue to pass and send to the fourth detector and to the third detector, which is a little bit depicted like that. So the movement of this this phi tube. Can you still hear me?
Yeah.
Hello. Oh, okay.
Yeah.
Okay. It seemed that I was signed out. Yeah. So the light coming from your sample will be very efficiently and very fast transmitted, and the sequel will reach all these detectors. So this is a very smart combination of different dichroic mirrors and filters. This was another fixed filter. Yes, we will have a fast simultaneous acquisition. Now, another option is to use so-called diffraction grids. And the principle behind, here I will start showing how it works in the Nikon system. So the emission light coming from the sample will arrive to a so-called diffraction grating, which it's composed of very small slits and grooves that are here in this diffraction grating, and which will allow the light will then be split. Then after it's reached this diffraction grating into its rainbow, like in a rainbow light in its components. And for the Nikon system, then after that, it will reach an array of 32 detectors. What we will get here, this is how it looks in the Nikon system. We have the diffractive grating, and then we have our 32 PMTs, which have a spectral resolution with the 32 detectors have a 10 nanometer spectral resolution, basically covering 320 nanometer of the visible spectrum. So then the emission light this ribbon of the light from your sample will reach these PMTs. What you will get, the signal will fall on each of these detectors, and each of these small detectors can be treated separately, basically could be treated separately, and it will give you an image with the 32 channels. Alternatively, what you can do, you can pull together some of these detectors to, for example, one larger detector. Well, you will with the help of so-called variable width slits or virtual filters, as they are called in Nikon. For example, if you want to image on the green your emission light, then you are pulling together several, and you will the green will image will only detect the green emission, or you can do the same for the yellow or the red, depending on your of the fluorophore and what you read often in your spectrum, that it's more convenient to avoid, for example, bleed through. Zeiss came out with also use diffraction grating in their system. Zeiss have they have altogether 34 detectors. In comparison with Nikon, we have had 32. But the principle how it works is the same, in the sense that the emission light coming from the sample will reach this diffraction grating, and it will split in this rainbow component, which then will be sent either to a central 32 channel gas detector, which is flanked by two other PMTs, one for the blue fluorophores and one for the red and far red fluorophores. And how the light is sent to either detector or one or detector or the other is with the help of so-called wedge prisms, which can be moved in one direction or the other, thus sending the light either to the blue detector or to the central detector or to the far red detector. This is the same principle. It's also in the LSM 880. Here we have an extra detector, if we can call it, which is for the every scan. Is exactly the same principle. Or the LSM 980. Here we have 36 detector. We have the 32, so the main 32 gas detectors, the two, one detector for the blue, the red, the near infrared. We have also, and the Airy scan. So again, the advantage is fast simultaneous imaging, of course, as long as you don't have bleed-through. Olympus, they also, of course, they have microscope, very good, and they also have employed the principle of splitting the light in a rainbow, also using diffraction grating system, and on top of that, what they do, they have a so-called volume phase holographic diffraction grating. That means that they angle this grating, which means that when they angle it, they will send it to different parts of this special mirror, which in turn will send the light to different parts of to different detectors. They usually have between 2 or 4 detectors, and exactly which part will reach which detector is also determined by so-called adjustable slits that they are, or before they were called widges. They call it slits. So as you can see, the principle all between this manufacturer or microscope is the same. You have a grating that will split the light, and then each of these manufacturer find a way how to afterwards to send the light to each of the detector, to the detector they have. Now we have also, we didn't talk about Leica, which is uses a prism to split the light in its rainbow components and then send it to its detector. They have 5 detectors. And for example, Stellaris, this is an image of how it looks in the Stellaris 5 scan head. If we zoom in a little bit, again, we see the light, the emission light coming from the sample will reach the prism, and then afterwards it will be pass it will reach either of the detectors, again by being sent and guided to each of the detectors using spectral sliders and different mirrors. So each of the light will reach its own detector with the help of the spectral sliders. It's very fast. Again, the very fast imaging. We can image simultaneous fluorophores when we are if we don't have bleed-through. So in conclusion, variable dichroic mirrors, variable filters, or diffraction grids, or CRISPR technologies, they have a lot of advantages. First of all, they give you great flexibility. You can adapt the width of these filters to the spectrum of the fluorophores, so you can combine several if you did your homework and you find out what are the overlapping parts, then you can move this so-called virtual filter, so you can avoid imaging, you can avoid the bleed through. If you have a very dense sample, you can widen the range of the wavelet that will participate to each channel, so you will be able to collect more light. Exactly, you can do the opposite. You can narrow down the range of the corrective wavelength to avoid bleed through between overlapping channels. But there are also disadvantages, and probably the main disadvantage is that the equipment is much more expensive. So now a few words about spectral mixing, because whatever, we do our best. We also want to do a lot of things. We want to use a lot of fluorophores. And quite often, whatever we are doing, even with this flexibility we have with the dichroic, virtual dichroic mirrors and virtual filters, there are occasions that we will not be able to separate our fluorophores just like that. And this is when we will perform. We would also utilize the power of the spectra by performing spectral unmixing. So here I have an example. Then we have two fluorophore, Alexa 488 and GFP. As you can see, their emission spectra is overlapping quite a lot. There is no way that we could really separate the spectrum, the emission spectrum between them. So if we will take an image, this is how the image would look like. And maybe even if you know the biology behind, where you will have the localization of the proteins which were labeled with the GFP or Alexa 488, it will be still very hard to or to prove or to show really what they are. So this is when we should or we have the we should perform or we can perform linear unmixing, as it's called in the Nikon calls it, or online fingerprinting, like is depicted in the Zeiss software. So when should what are the applications for unmixing? Well, when we are using a fluorophore with very strong spectral overlap, if we are using many fluorophores, if we want to reduce the autofluorescence, or we want to avoid time-consuming sequential imaging. So let's assume that we have a sample where we have 3 fluorophores. The idea behind spectral mixing is that what we have to do is to tell the software where our fluorophores are. So the software, by applying an algorithm, is able to remove the fluorophore, the spectrum, the fluorophore where they don't where they do not belong. And to do that, we need to extract the spectra from our sample. So the first step, that's why I said if we have 3 fluorophores, we will prepare 4 samples, 5 if we want to get rid also of the autofluorescence. And in 3 of our samples, we will only have 1 fluorophore at a time. Let's say we have blue, green, red. In one sample, the same type of sample, you have 1 fluorophore, the second, and the third fluorophore. And we take an image and we extract the spectrum from each of them. So we will have the pure spectrum of our fluorophore in our sample, but only by itself. In the step 2, we take an image of our real sample with all the fluorophores. As I said, if we have a sample which is for our negative control, with a sample that we make for our autofluorescence, we'll also image that sample and we will extract the spectrum of the autofluorescence. In the step 2, we take our real sample with all fluorophores all together, and we take an image, and this is the image, for example, how it can look like. All the since we have bleed through between all our fluorophores, so we cannot really know which the contribution of each of the fluorophore in each pixel. But what and since we have a spectral detector, as I said, we can treat all these images that fall on the little one of these the 32 detectors as its own image, and this is how it will look like. But now, having the information how the reference spectra looks like, we can apply an algorithm which will determine the relative contribution of each of these fluorophore for every pixel of the image. What is the probability that it will this fluorophore would be in that particular pixel in your real image? And after performing the spectral mixing, you will get the unmixed image. And that's how spectral mixing is done. So now, if you look at we look at our sample, the first example, and we have GFP and Alexa 488. If we would perform spectral mixing, now we will be very clearly be able to discern where Alexa 488 and the GFP signal are. There are also, you could see the advantage of using spectral mixing, but there are also this limitation of using this technique. It's not always easy to acquire the reference spectrum, especially if you have you work with transgenic mice. If you have too big of a differences in the brightest in your fluorophore of the sample, you will get very weird results, not really trustworthy. And in my workshop, I think that I give a very good example of what can happen. If you really have fluorophores with the spectra that are virtually superimposed or only having a very few nanometers between them, it would be very hard to perform unmixing. Thank you for your attention.
Thank you, Gabi.
Any questions?
So, yeah, I would like to know. Yeah, okay, Robin, go for it.
Yeah, so maybe I didn't catch. So how do you know if you have a spectral detector? Which one? Was it only the grid one, or was also the prism spectral detector, or how do you know if you can actually do this last step?
Well, first of all, you have the specification in the at the microscope that you have a spectral detector. Like in the Zeiss, they have the spectral detector. The Nikon has a spectral detector, and then of course you can do, because you also need to have the software available to do the spectral mixing. As far as I know, you have to have the specification that you what kind of detectors you have.
Yeah, because I tried to Google now the Leica that I have. It says SP detector or spectral detector, but I have never seen this in the software. But maybe I just have to look then, I guess.
Yeah, but it might be that it's a special module software that you need to purchase.
Okay. Yeah.
But I think that as long as you can sort of move the filters, let's say, the width
Yeah.
of the filters, it means
Yeah.
you have some sort of a spectral detector.
Yeah.
And therefore you should be able to do unmixing.
Okay.
With the correct, if you have the correct software module as well.
Yeah, I'll check it out.
I'm pretty sure that you can, but I mean, the whole idea is here that you can move and you can narrow it down to very small nanometers, so you can really resolve what you need. So, but yeah, you definitely need a module for it. But as far as I know, Zeiss and our Nikon systems, the more modern they have. But I think even the Leica has, you can perform spectral mixing with the Leica system.
Yes, Andres?
I had a question. The Leica microscope I use has a functionality where it can do photon counting and delay from excitation, I think, to differentiate between autofluorescence and a fluorophore. Is that something that is where you need a spectral detector, or is this another system or another special detector in the microscope?
I think that is a different functionality that you have with Leica. And right now I don't remember the name, but that's not for spectral mixing, if that's what you asked.
Yeah, I was just wondering if,
But it's
just out of curiosity, if that's had something to do with the spectral aspect of it or not.
No, it has to do with the detectors that you have. So these detectors, the high D detectors,
Yeah.
they are photon counting detectors, basically. So you can do photon counting, but it means that you can also so I very briefly mentioned FLIM, and we have several people here who work with FLIM as well. But basically, additional when you talk about fluorophore, additional to excitation, the excitation spectrum and the emission spectrum, we also have a lifetime. So this is a characteristic of the fluorophore as much as excitation and emission spectrum. And so the lifetime is like this. When we excite the fluorophore very, very shortly with a very short pulse of light, like a femtosecond pulse of light. So we turn on the light, turn it off immediately. The fluorophore gets excited and then it fluoresces, but then it slowly stops because there's no more excitation light. And this curve of getting exciting and slowly slowing down, this is typical. This is a signature, let's say, of the fluorophore, and it takes nanoseconds, let's say. But the shape of the curve also is typical, so you can recognize fluorophores based on their lifetime, even if they have the same excitation and emission spectrum. Plus, the lifetime changes based on the environment. So if you have one fluorophore that is in the cytoplasm and then it gets into a vesicle, for example, where the pH is different, then the lifetime is going to change. So even though the excitation and emission spectrum have not changed, you can differentiate if it's inside the vesicle or not based on this lifetime. Now, the HD detectors, because they are photon counting, they're able to basically count each of the photons that come when you do this pulse of light. And so this is the principle of FLIM. And this is the reason that, because most of the autofluorescence in the sample has a short lifetime, they can make a pulse of light and let the first photons go without acquiring. And then they start the detector with a little delay. It's a tiny delay of maybe 1 nanosecond or something like this. Very tiny delay. And that means that they basically get rid of the autofluorescence this way. It's a nice trick.
Okay, that's a very cool trick.
Yes. But it's nothing really to do with the fact that it's a spectral detection. It's more to do with the detectors themselves.
All right, thank you very much.
Yes. Who is first? Was it Shay or is it Ellie? I forgot.
Maybe I go first.
Okay, go first.
Thanks. I have a question about spectral. Oh, sorry. Am I muted? Yes.
No, you're not. We hear you.
So I have a question about spectral mixing. Is it possible to have a single stained fluorophore beads to use as a reference control? That particularly if
you can, but it's not really advisable. What you should have is to have exactly the same, secondary or fluorophore that you have in your sample. So that's, and that maybe I should have mentioned. It's not like you have to do this, to get your reference spectrum. So to prepare all this sample all the time. You do it once at the beginning, and I have this sample, I use these fluorophores, I don't know, all the Alexas. and you make it once separately with your sample exactly in the same way as your real sample. You do it one and you extract the spectrum from your sample.
Okay.
And after that, you can use this spectrum over and over again, as long as you don't change. You changed already the tissue or something, the tissue, then you should redo it. I mean,
Yes.
all this software that even they put the spectrum of all these fluorophores is there, so you could actually use, but it never gives you the same result. So it's better to do everything with your own sample. You do it once, you save the spectrum, and then you are done. You can reuse it for that particular application over and over again.
Okay, thank you.
Ellie?
Yes, because I am a bit confused with all these components like the diffraction grid, the prism, and the different systems. Could you please repeat, like in a generic way, how the emission light is going inside the detectors?
So basically, let's say if we are using the prism, you get the light and the prism. So what happens with the light, then it encounters the let's say the prism, the medium. So you have this is a Snell law, it will bend, it will bend once and then it gets out from the prism, it will bend again. And then it will do that, it will make like a rainbow of light. So now different manufacturers found, and this is exactly how this diffraction grating is working in some way. I don't know the technical details, but the point, all of them will give you this rainbow, so the whole spectrum of the light. Now the question is, how is this light guided towards each of the detectors that you have? So, for example, what they do is that you have all your detectors, like for example, let's say LSM 880 or 980. Then what you do, you have your spectral, the 32 gas spectral detector, but that's probably only 320 or a few nanometers. So you want to have some extra detectors that they catch the rest, the blue and the far red. So, and then we have this the like wedges that you say, oh, now my light is come. So I guide the light that comes. We can move these wedges, and you say, I guide this light to go towards that detector. The next one, oh, I want this light to reach my gas 32 spectral detector. Or you guide by moving, or it's not you moving, the machine is moving depending how you made the setup, it will guide to reach different detectors. Probably like in I have also some extra mirrors that will help you to guide the light depending how these wedges are putting how far away to guide the line.
So Ellie, which system are you using?
The 980.
980. So you do have this detector with 34 detectors. 32 in the spectral, +1, this is what you have.
So the
Yes,
one
I could see these. I could see these PMTs, but I didn't know that they are used together with a spectral one. So I was confused.
Right. Yes. You can. You can have that. Basically, they're used, but what do I say? You're going to use one. So these. Yeah, I mean, they are used as a 34 detector. You can also use them individually in different tracks, but they are used as 34 detectors. And you know that in the Zeiss software, you can click on the little plus to the spectral detector that is in the middle is called channel S, right? You have the channel 1, channel 2, and then channel S. There's a little plus there. When you click on that plus, you can break it into several blocks as well.
On top of that.
Yeah. So you could make several channels. You could make 4 channels with channel 1, channel S1, channel S2, and channel 2.
And this is when you are doing sequential too, then you don't really, you don't have a choice, and then you want to image the fluorophore maybe separately, or you can combine certain fluorophores.
If you're a bit unsure how to work with the spectral detector in ZEISS, I would really recommend going online and asking, looking for a video to show you how to use the spectral detector, and then you get really nice tips of the stuff that you didn't get during your training. Then you combine it that way. Yeah.
Yeah, thank you.
Good. So thank you, Gabi. And if there are no more